Okay. So in this lesson, you're going to look at how DNA sequencing of the DNA from two different organisms allow us to know the evolutionary relationship between these two different species. Okay. So this is an example of the evolutionary tree of a human to show how closely related we are from one another. So you see the French and the Han Chinese, right? So the French and Han Chinese, they are different races, but they have got a common ancestry in a quite recently as compared to with other races, isn't it? Correct? So this is called evolutionary tree. But then when you trace all of them backwards, you can find that all of them also got a common ancestor called Homo sapiens. Correct? But you trace further up, actually Homo sapiens and this, what is it called? Neanderthal. Okay, Neanderthal is already extinct, no more here already. Both of us got the common ancestor, but you can see that both of us are very different because their jaw is very big, okay, very big and long compared to the current Homo sapiens. Our jaw is actually very small. You can see the spacing inside is actually very small. This has to do with what we eat right nowadays, right? They have to have very big jaw to eat all the huge meats that they hunt, isn't it? And maybe sometimes raw, right? For us, we don't eat meat, we eat fish ball. We eat burger. All are very soft and highly processed. So we don't need to have all the huge muscular big jaw to chew them, right? So therefore, our jaw becomes smaller and smaller, okay? But our teeth still remain that big. All right, so we want to identify evolutionary relationships between two different species. How do we do that? Is by finding genetic relatedness between organisms. Okay, that means how closely related we are has to do with how closely related our DNA are, isn't it? Because we started off with the same DNA, the ancestor, right? Same DNA. Then, because of natural selection, different alleles are being selected, right? So the allele frequencies start to change. So as time passes by, our differences become more and more. So based on the differences, we can see at which point should we draw our common ancestor? Is it nearer or further away? So it's based on genetic relatedness. So we want to find out genetic relatedness. Of course, these two, they got more DNA that are common. These two got less, this is even less, right? So this is to do with genetic relatedness to tell us how recent is our common ancestor, how recent do we actually start to diverge away from our common ancestor and subjected to different selection pressure. Okay. So there are two methods that you learn in your syllabus. The first one is genetic fingerprinting. And the second one, which we're going to learn today, is DNA hybridization. So these two methods will allow us to find out genetic relatedness between two species, and hence we're able to draw the evolutionary tree from that. I am guessing that one of these days, they're going to ask a student to draw the evolutionary tree in exam. You will not find any past year question because they have not asked before. So that means they can ask, right? They will ask, but I do not know when. Every time I predict, it'll come out 5 years later. So tell your sister or brother 5 years later. Now this is genetic fingerprint, right? So we already know that for organism A, B, C, D, E, or individual A, B, C, D, E, their genetic fingerprint is different because they've got different number of bands in different locations. So it's unique to them. But as long as you've got the DNA that are DNA that are very close to one another or similar in base sequence, similar in length, right? Then the band will be located in the same location, isn't it? So the more the bands that are same, the higher the number of bands that are in the same location among those fingerprints, the higher the genetic relatedness. For example, you see between A and B, right? How many number of bands that are similar in locations? Quite a lot, isn't it? Right? These two are the same, these two are the same, these two are the same. So when the more similar is the location of the band, it shows that it's more related to one another. That means the common ancestor is more recent, isn't it? The timing for them to diverge from one another is less. That's why they have got more similarity in the genetic makeup. And because of more similarity in genetic makeup, that means phenotype also quite similar, right? Because VP is equal to VG plus VE, right? Okay, all right. So, second method is called DNA hybridization. What is DNA hybridization? So we take the DNA from the two species, species A and species B, and then we apply heat on them to denature the DNA. So that means we're going to break a hydrogen bond, split them open into single strand. Then we put those single strands together so that they will form complementary base pairing. So we will reduce the temperature, we mix the DNA of the two species, and we reduce the temperature so that they allow hydrogen bonds to be formed. So of course, when the bases are complementary, they can form hydrogen bond. Whichever part that is not complementary, like those red color crosses, then there will be no hydrogen bond being formed. So after this process, this piece of DNA will be subjected to denaturation by temperature. So based on the temperature required to denature, then they will know how closely related is these two strands of DNA. So the higher the temperature, so let's say, for example, you need higher temperature to denature, meaning to say they have more or less hydrogen bond. More, right? So more hydrogen bonds. So more hydrogen bond means what? More similarity, right? More bases. More complementary bases. That means more similarity in the DNA, isn't it? More similarity means they are more closely related, right? So therefore, common ancestor is more reason or less reason? More reason. So divergence is more or less. So less divergence. Less divergence means what? Less time for them to evolve in different selection pressure. Correct? Less time for them to evolve in different selection pressure. Can understand all this? This is how we find out whether the two species are related, are closely related or not. Okay. So when they do this DNA hybridization, the DNA that they use is actually mitochondrial DNA. So they use it to compare similarity in the base sequences of the mitochondrial DNA, which is called mtDNA. I think some of you want to copy something, is it? You want to copy this, is it? Am I still recording? Yes. So why do they choose mitochondrial DNA to do the DNA hybridization to compare the genetic relatedness between the two species? So there's a reason behind that, okay? So to compare the similarity between base sequences of mitochondrial DNA, which is mtDNA between two species, why we use this? That means in DNA hybridization is used to compare similarity between base sequences of mitochondrial DNA between two species. So why we choose mitochondrial DNA and not the nuclear DNA? Okay, because human mitochondrial DNA comes from female line. Come from female line means the mitochondria that we're carrying in our body comes from the ovum of the mother. And from the mother, it comes from the ovum of her mother. It's all from the ovum only, okay? Because why? Only ovum contains mitochondria that they pass on to the zygote, and from zygote becomes embryo and fetus. For sperm, after fertilization, the mitochondria of the sperm will be destroyed by the ovum. sperm got carry mitochondria. When it fuses with the ovum, right, the mitochondria of the sperm will be destroyed by the ovum. So this mitochondria from the sperm will not be passed to the offspring one. So whatever mitochondrial DNA we have in our mitochondria, right, they are all from mother's lineage, the female lineage only. Okay. And all the mitochondrial DNA in our body, in all the cells, are genetically identical because they are all produced by DNA replication. Can or not? It can originate from one zygote, right? When the zygote divide, then the mitochondrial DNA also divide. So they are all genetically identical. Okay. And mitochondrial DNA is circular. Circular, what's so good about circular? Circular DNA, there's no crossing over. Only chromosome got crossing over. So when there's no crossing over, we will not introduce any genetic variation, right? Correct or not? When there's no crossing over, you will not introduce genetic variation. Crossing over causes exchange of genetic material and produces recombinant type. So here is not recombinant type, always passed on from generation to generation as it is. So the only way that it can cause a change in mitochondrial DNA is because of mutation. Only mutation can cause a change and nothing else. And then they say that mitochondrial DNA mutates quite fast. Why? Because they are not protected by histone proteins, the chromosome. So you know that whenever a molecule is protected or surrounded by another molecule, the another molecule will be holding it so that it will not easily change in structure. But because mitochondrial DNA is totally naked, it's not surrounded by the histone protein, it can easily mutate. And then some more mitochondria carry out oxidative phosphorylation. So a lot of oxygens are there. Those oxygen can easily oxidize and change the DNA structure. So therefore, mutation can occur quite quickly. So because of higher rate of mutation, mutation will give rise to new allele. Therefore, there'll be more variation in genotype. Correct? Different allele means you got more different allele, more different genotype, right? So therefore, there'll be more variation in phenotype. Therefore, because mutation is faster in mitochondrial DNA, so it can allow us to easily measure the differences between two organisms very easily from generation to generation because there are changes very quickly. So if there are no changes from one generation to the next generation, even though they are different, but we can't measure them. Okay, so that's the advantage of this. Because it mutates very fast, from generation to generation, any slight changes we can see very quickly. So we can measure the differences very easily. Okay. So this diagram shows the lineage of two things in a human. For male child, all the Y chromosome, the DNA of the Y chromosome is all from paternal lineage. That means the Y chromosomes come from father comes from his father comes from his father and his father. Whereas for the child's mitochondrial DNA, it only comes from mother, so it's from the female lineage. Because from the mother, she received from the grandmother. Grandmother received from great-grandmother. So it's all female lineage. Okay, so human mitochondrial DNA is from female line. Line means what? Line means lineage. Okay, that means you trace back who is the mother of this one, and after that, who's the mother of the grandmother, who's the mother of the great-grandmother. So that lineage, okay, where they pass down the gene. So after we have done the DNA hybridization to find out the degree of similarity and differences between their DNA, we can use it to draw the family tree in a family tree, evolutionary tree. So this example of evolutionary tree of human. So let's say, for example, we found out that, okay, between human, chimpanzee, and gorilla, the degree of genetic variation is very little. It's about 10. Let's say, for example, it's 10, 10, 10. So the degree of difference is 10, 10, 10. So that means to say all of them have got a common ancestor, right? In a recently. So all these three, because the differences in their DNA is 10, 10, 10, therefore they will have a common ancestor in the recent timing. As compared to chimpanzee, human and orangutan. So let's say, for example, the differences in our DNA between human and orangutan is, let's say, 15. That means to say, our common ancestor between orangutan and human must be further away from this one, right? Right? It's not so recent, it's further away. So that's why you have to draw it this way, further away. So this is common ancestor. So in that way, you can draw the evolutionary tree based on genetic relatedness, based on how recent is the ancestor. The more related, the more closely related their DNA, the more recent is the ancestor, the less timing for them to diverge from one another. Okay, so that's how you draw it. Okay. So second example we're going to look at is genetic differences between anole lizard species in the Caribbean by DNA hybridization. So they found that you got this lizard, which you can find in Cuba, in US, in Andros, and Ecklins. Okay, so they want to know that, okay, so all these species are different. Where do they come from? Which is the common ancestor? They cannot know until they do the DNA hybridization. Okay, so after they did DNA hybridization, they got this data. So the value in this table shows the degree of divergence of their DNA from one another. So what does it mean? So you see A. brunus and A. smaragdinus, right? The degree of divergence between their DNA is 12.1. Whereas between A. brunus and this one is 16.7. So this one is more different, right? The DNA of this with this is more different than this with this, right? And then this 11.3. That means the DNA of this is more closely resemble the DNA of this than these two. Okay, so this value here measures the degree of divergence. That means the differences. Okay, now based on this one, there's something that we can see is that for all the three species that we can find here, all of them are more closely related to A. porcatus. Can you see that? Okay, you see this one, all of them, they got less differences with A. porcatus compared to the rest. So they're more closely related to porcatus. That means to say all these three could have evolved from porcatus in Cuba. So they could have all originated from porcatus in Cuba. And because of certain incidents, it caused them to spread to these three different locations, maybe because of hurricane. Okay. So because of that, they are still very closely related to this original species. Whereas they are very different from one another because they are subjected to different selection in these three regions. Right? In the island over here and here, the climate, the weather is different. Therefore, selection pressure is very different. So all these three are subjected to different selection pressure. So they are different from one another, but they're still very similar to the original ancestral population. Okay, that's why you see this one is very different with these two, but it's more similar to this. That means A. brunus is located here. He's very different from this and this, but still very similar to this. Okay, so this is how we can look at the data and actually explain things, how it works. So therefore, these three species, they have arisen from the original ancestral species in Cuba. And this one looks like, is it allopatric or sympatric speciation? Allopatric speciation, right? So this looks like allopatric speciation. Okay. So let's do two partial questions. Let's do two partial questions. First one. This is June 07, paper 5, question 2. Why paper 5? Why not paper 4? Paper 5 is when you got the data like this one here. Genetic fingerprint is the data. They ask you to analyze the data and do conclusion from data. That one is paper 5, right? Analyze the data and do conclusion. So let's do the questions together. A, explain how the genetic fingerprint provide evidence that these mammals share common ancestor. If they share common ancestor, that means they must have got some DNA that are similar to one another, right? That comes from ancestor. So let's see, are there any band that are similar to one another across all these 5 organisms? I think got, here got, right? And then, so it's a 2-mark question. So first mark, you need to tell that how you see. You see that they have got the band and the same location for all these 5 organisms, 1 mark. Then of course, because it's a data, there is a reading one, right? So you have to give me the reading. So what's the reading? Reading is you have to tell them, so what is the length of this DNA in this location? So what is the length? So the length is between 500 kb and 750 kb, right? So I will write between 500 kb to 750. If you are more hardworking, then you can go and find out. So this one is how much? I think it's closer to probably 640, 50 like that. 30, 40, 50, around there. Okay. So that's the 2 marks, right? So let's look at the mark scheme. So the DNA fragments of similar sizes. So you can find DNA fragment of similar sizes or similar position of the band. Okay. And then refer to location 400 to 700 kb. That means you give me any value within this range is correct already. Okay, next question. Use the information in the diagram to explain the evolutionary relationship between these mammals. 5 marks. That means use the data that you have to do conclusion. So let's go back to there and see how to do conclusion. We are supposed to talk about genetic relatedness, right? So let's look at between A and B. How many bands are exactly the same? Is this same or not? Is this same or not? Is this same or not? Are there any more same one? No one, because this is higher than this, right? This is higher than this, even though it looks very similar. So I see that there are 3 bands. For between A and B, there are 3 bands. They are located in similar location, right? And therefore similar length or similar size. So this is what I see from here. But because I need to get evidence, I have to give the length. So the length is this one and this one and this one somewhere here. So therefore you have to give the 3 reading, right? So this one is probably around 300. Okay, around 600, 20, 30. This one around 850. 850. Okay, so that is for this part here. So let's look at the mark scheme. A and B are more closely related to each other because they've got 3 bands in common, and the location of the band is around 300 kb, 55 to 6,000. I don't see there's a 6,000 there, right? What? 550 to 600 kb, is it? And 800 kb. Okay, and then the conclusion is not complete yet. That means they must have separated or diverged from one another from the common ancestor recently. Okay, so they must have separated most recently in evolution, or they must have a common ancestor recently. Okay, so this is first, second, third is together. Now, what about E, C, D, and the rest? So let's look at E, C, D, and the rest. E. Hey, what am I doing? Okay, next. What about ECD? So let's examine ECD. Are there any common banding between each other than only the one that we have identified, this one? So this one is common between all of them. Other than that, I think no more line that common, correct? So that means between C, D, and E, they only got 1 common band. So that means to say that 1 common band. So the common ancestor is less recent, isn't it? Okay, they only got 1 common band. That means they are less genetically related. So the common ancestor is less recent. That means they are subjected to longer time of divergence. Correct? That's why they are more different. Okay, so this is C, D, and E. They also said between D and E, how many number of similar bending pattern? Only one, isn't it? Can you see not? Can you see? Between D and E, there's only one. So D and E are not so closely related to one another because they only got one band that has got similar location. Okay, and then you say, what is the band? So there are many ways for you to read. So let's look at the mark scheme. What other ways they have read the chart? Okay. A and B are closely related to each other, 3 bands in common. Just now I saw 5 bands. Okay, you see again, you see, okay? So what are the number, the length of each of the band? And therefore, because of that, they have separated most recently in evolution, or you say that their common ancestor is more recent, also can. Okay. E is distantly related, has from D because they only got one band that's similar. So E is not closely related to D because it's only one band. That means E and D have separated long time ago in evolution. Their common ancestor is long time ago. Okay. So that's why I said separated early in evolution. What about C and D? C and D are closely related with each other because they got 2 bands in common. So you check the bending pattern, is it 2 bands in common? And then statement followed by evidence from the data around 700 and 800 kb. So this is enough for you to get 5 points already. The rest are also correct points. If you can see, A and B or C and D are more distantly related, separated in evolution. That means this group A and B and C and D, A and B are very close to one another, right? Okay, so that means they are very recent in their common ancestor. So this is A and B. I got a feeling that one of these days they're going to ask you to draw the evolutionary tree. Definitely, but I do not know when. Because there's no question in past year that asks a student to draw, but because from here you can draw, I think they will ask you to draw. So sketch lah, okay? So if we sketch, it means A and B is here. C and D are not closely related, right? C and D only got one band similar to A and B. That means C and D common ancestor with A and B are very, very long time ago, C and D. So their common ancestor must be far, far away. Okay. C and D got a common ancestor long time ago, which is similar to A and B. So all of them got one common ancestor at the same time. Because all of them only got one band similar, right? All of them only got one band similar. That means all of them only got one and similar ancestor long time ago. So that's why I joined all of them together to one common ancestor based on the fact there's only one band similar. Okay. D has common band with A and B. C has common band with B and D. D has common band with A and B. Okay, fine. A has common band with B and D. Okay. So this is to just show what is the common band location. Fruit fly, yes. Drosophila has many different species. So over here, they say there are 3 species, and these 3 species are found in different locations in US. This is where they are naturally occurring. So you've got 2 different species, okay? Sorry, 3 of them. So what's the question? So they say that they have done DNA hybridization. Okay, the base sequence of 4 regions of DNA have been sequenced. The divergence of this base sequence in all these all these 3 species was calculated. So these are the 4 DNA regions. So they are actually taking these 4 regions and try to do DNA hybridization among all these 3 species. And then this is the value for divergence. That means the differences. The value for the differences. That means the larger the value, the more the difference. Okay. So what does the question ask the student? With reference to figure 5.1, describe the evidence that D. mirinda may be more closely related to D. persimilis and D. pseudoscura. More closely related means this value should be higher or lower? Higher or lower? Lower. So are they all lower? So D. mirinda, this value is lower for DNA 1, DNA 2, DNA 3, DNA 4. They're all lower, right? So therefore, from the table, you can find that the percentage divergence of base sequence of persimilis is lower than pseudoscura towards D. mirinda, correct? So this will be the answer. But then because it's 2 marks, you need to provide evidence from the table. That means you can say that, okay, for DNA number 2, it is 7.3 for persimilis, but 8.1 for pseudoscura. Okay? Can I? So put your statement and then give your evidence. So every time if there's a table and there's a graph for you to describe something, you have to quote the figures. Okay, last question. Suggest why there is more divergence in some regions of DNA than in others. Why is it some DNA got higher divergence, like 7.3 is more divergence, 8.1 is more. So DNA number 2, there are more divergence, more differences compared to DNA number 3. So why is it this one got more divergence than this? What can cause the DNA to be different? Mutation. Very good. So how mutation is correct, but this is 2 marks. What is the second mark? Why some mutate more, why some mutate less? Correct or not? Because you said mutation causes a difference. So the more different one is because of what? The less different because of what? Okay. Okay, so some regions of DNA are more prone to mutation than the other. Why? Because mutation, right, can occur anywhere in the DNA. Let's say if this is a chromosome and this is a gene, gene of the chromosome. Mutation can occur in the gene and mutation can occur outside the gene, right? Now, if the mutation occur outside the gene, is it very serious for the organism? Outside the gene wall? No, because it doesn't code for anything, right? So will it affect the organism? No, right? So therefore, will the organism survive? Will it pass to offspring? So this kind of mutation that occurs in a non-important region of DNA, it can be passed to the offspring, therefore you can measure them. But the moment the mutation occurs in a gene that's important, let's say gene for aerobic respiration, you change in the base sequence, change in the protein structure, cannot catalyze the reaction, cannot carry out respiration, organism die, right? Will this mutation be passed to the offspring? Cannot. Will you see this in the offspring? So will this section of DNA will have got more divergence or less divergence in offspring? Less, because you can't find them. Any changes at all, they die. So it's not passed to the offspring, you cannot measure them. So the divergence in the gene area will be less than divergence in non-gene area. So therefore, look at the mark scheme. Some mutation in some regions are likely to be fatal. Fatal can cause death, especially in the gene area. So therefore, there tend to be less divergence because it's not passed to offspring. Okay, because it's located in important gene, so they cannot survive. It's not passed to offspring. Why? Because this gene will code for essential protein. Once mutated, protein changes shape, function cannot happen, organism cannot survive, they die. Okay, so that's all for today's lesson about this DNA sequencing, how DNA sequencing can show evolutionary relationship between species. All right, so your homework is draw the evolutionary tree. I also have to go and draw and see. Okay, so that's all for today. All right, bye everyone. Okay. Have a good weekend.